To ensure survival, our planet's ecosystems are characterized by struggle and displacement. Potential enemies must be identified and fought off. Food sources must be developed. When animals settle in new areas, their long-term existence is not predetermined. A struggle with an uncertain outcome begins. In the EU-funded REACT project, researchers are working together to improve pesticide-free pest control for invasive species that threaten agriculture and trade. The pest species addressed by the project are Bactrocera zonata, the peach fruit fly, and Bactrocera dorsalis, the oriental fruit fly. Let's look at the methods used to understand the ecological impact of these fruit flies that are not yet endemic in Europe, but have recently started to appear in southern European countries. To protect the biodiversity of European ecosystems, it is important to understand all the effects that a particular pest can have on the balances in cultivated and uncultivated refugia. First and foremost, this means identifying a list of plants and testing their host status for both fruit flies under laboratory conditions. But let's hear from Hélène Delatte, an etymologist from the French island of La Réunion. She knows more about the impact both flies could have if they reach Europe.
The two species will be coming in a complex environment, a different ecosystem that they know. And in this ecosystem, you add two new species or one new species, depending how many will invade. So the ecological equilibrium that we have now can be changed. So if these species decrease in numbers, so all its predators, all its parasitoids, all anything in the food web chain that will, in a different trophic level that will eat these species, used to be, will not have any more anything to feed on. So you see your equilibrium will change. So that is the first direct impact of the invasion on the arthropod, which are all the other insects and acarians that are around. But you also have higher impact, like indirect impact, because this insect could be eaten by birds, for example. So the fruit fly are frugivorous. So they, especially Bactrocera dorsalis, is attacking fruits when they are very, very green. And so they will not let the fruit going to ripe sometimes. So you won't have seeds for the next generation, especially for wild indigenous fruits, which are in our landscape, in European landscape. So these fruits, these plants will miss a generation.
The impact on natural ecosystems in Europe, including grasslands and forests, but also agriculture, is just one dimension that needs to be tackled. On another, literally microscopic level, the fly itself harbors a great diversity of species that influence its fitness. George Tsiamis, professor of environmental microbiology, explains the research carried out to better understand the insect that is itself living together with other species and organisms.
We try to see the insects as a holobiont. And what we mean as a holobiont, we look at the insect, but also other organisms that they live inside the insect. We try to understand the role of these microorganisms that they live inside the insect and how these microorganisms affect the growth, of these specific organisms. Because we do know, for instance, that they can change and alter the number of eggs that they lay. We know from other examples that the time that an insect needs to develop changes when some specific bacteria are living in an insect.
In the context of pesticide-free pest control based on sterile insect technique, it then makes sense to collect further parameters, which relate to cohabitation with other species. Sterilized male flies must not only survive under certain nutritional conditions, but after all, also mate. Avgoustinos, a researcher and expert in rearing and release mechanisms, explains the competition between flies in the lab and in the field.
Our main question is what the sterile males that we are going to release, what they will actually do in the field, how they will perform. And this is what we consider as the biological quality of these males. So that means that they have to be able to survive and mate and being antagonistic with the males of the natural population. So in the first set of laboratory experiments, we can check their flight ability in specifically designed flight meals, their survival under stress conditions, and also their survival or longevity under no stress conditions when we provide them food and water. So this is how we extrapolate some information about the quality of our strains. Later on, when we want to go really in the field, we have to do larger scale experiments, open field or semi-field experiments. There we test actual parameters in the field, like dispersal of the flies. We need to monitor how much they can fly and how much they will survive in the field. And also in semi-field experiments, we can see if they are competitive to wild males. So we see how much they mate and if they manage to suppress the natural populations under contained conditions. Proper feeding and proper handling, proper transporting and proper way of releasing are the parameters that have to be very carefully checked in order to have good sterile males in the field.
As we see, it is important to understand the impact of a species on the existing ecological environment for several reasons. Assessing behaviors in different climates and diagnosing the perfect diet for a species helps researchers find alternative ways to protect our agricultural production and ecosystems as a whole. Thank you for watching.